What is the difference between reproductive and therapeutic cloning in genetics? Imagine being able to create a perfect copy of a living being or produce healthy tissues for medical treatment. Both ideas sound like science fiction, but they are real applications of cloning technology. Today, we're exploring how these two types of cloning differ and what they mean for medicine and ethics. Reproductive cloning is about making a new organism that is exactly the same as an existing one. It uses a process called somatic cell nuclear transfer. In this process, a nucleus from a body cell is inserted into an egg cell that has had its own nucleus removed. The egg then begins to divide and develop into an embryo. If this embryo is implanted into a uterus, it can grow into a fetus and be born as a clone of the original donor. This method has been successful in animals like sheep, cows, and mice, but it is banned for humans in many countries because of ethical and safety concerns. The main goal here is to produce a living, breathing copy of an organism. Therapeutic cloning also uses somatic cell nuclear transfer, but the purpose is very different. The embryo created in this process is not implanted into a uterus. Instead, it is grown in the lab for a short time until it reaches the blastocyst stage, which is about 5 to 6 days old. At this point, stem cells are harvested from the embryo. These stem cells are special because they can develop into many different types of cells, like heart, brain, or skin cells. The goal of therapeutic cloning is to produce personalized cells or tissues that match the patient's DNA. These can be used to repair damaged organs, treat diseases like Parkinson's or diabetes, or study genetic disorders. Because the cells are genetically identical to the patient, they are less likely to be rejected by the immune system. From a genetics point of view, both types of cloning involve creating an embryo with the same DNA as the donor. But what happens next is where they differ. Reproductive cloning aims to develop a full organism, while therapeutic cloning focuses on generating cells for medical use. Ethics play a big role in how these technologies are viewed. Beneficence, or doing good, supports therapeutic cloning because it can help treat diseases and reduce suffering. Non-maleficence, or avoiding harm, raises concerns about safety, possible abnormalities, and long-term effects. Autonomy involves questions about consent, especially when using human eggs and embryos. Justice asks who gets access to these expensive treatments and how they are distributed fairly. A major ethical debate surrounds the moral status of the embryo. Some believe embryos have the same rights as born humans, which makes creating and destroying them for research controversial. Alternatives like induced pluripotent stem cells, which are made from adult cells, avoid some of these ethical issues but do not replace all uses of therapeutic cloning. In practical terms, therapeutic cloning could lead to personalized medicine, where treatments are tailored to an individual's genetic makeup, reducing rejection risks. Reproductive cloning, if ever used in humans, raises questions about identity, family, and what it means to reproduce. Both methods challenge society to balance scientific progress with ethical responsibility, and ongoing discussions are needed to guide their future use. So, to sum it up, reproductive cloning creates a genetically identical organism, while therapeutic cloning produces cells that can be used to treat diseases. Both are based on the same genetic technique but serve very different purposes and raise different ethical questions.